Good morning or good afternoon, everyone. This is Jimmy Botella, and I am recording this in my little recording studio at home. This is a report on the field diagnostic protocols that we've been developing for Cassava on this ACR project. Let me share the screen. Hopefully, this will be recording. As I said, this is a report on the development of the field diagnostics that we have been working for Cassava. This is the work of myself, Mike Mason, and Jessica Hintzsche. Let's just start with it straight away. This is what we've done so far. The goals that we had at the very beginning of the project for the Cassava and diagnostic method is develop something that can be used in the field, that is easy to use, that uses minimal equipment, that it doesn't cost too much, and of course that is reliable, is robust. All right, but the truth is that we are also very interested in another set of goals. The other set of goals is we want to produce something useful, means something that works in the place that is intended to work, that is not Australia and it's not our lab. We want to produce something that our local partners trust and use. What do I mean? What I mean is, at this time, our local partners are using it because they're part of the project, but we need to develop something that it works for them, meaning that they trust their own hands and their own results. We need to produce something that is, they are comfortable using the method, meaning that it's not too much for them. All right. Also, we need to produce something that they participate in its development. There is nothing like having ownership of something. Otherwise, you're just doing something because you have a project and the project finishes and you don't use it anymore. All right. And that comes to the third goal. That is, we need to produce something that does not die at the end of the project. That is very important. Well, let's start with the optimization for the LAMP, for the Cassava mosaic virus, all right? The first thing that we did is, because we're using LAMP, LAMP is not like PCR. LAMP is a fantastic method, but the development of primers, the design of primers is quite complicated. So you always need to start with a number of set of primers. Some of them were available in the literature. We have found that, 80% of the time, things that are available in the literature, they don't work for us for whatever reason, but they don't. So we ended up designing quite a lot of different primer sets, and then we tested them one by one. Some of them were good, some of them were not so good. At the end, we decided 3 of the primers that were very, very reliable. The primer sets that we're talking about is what we call the A5, the B1, and the B8. As you can see here, this is a normal test where we have 1, 2, 3, 4, 5 healthy plants versus 5 infected plants, and you can see that there is no false positive and there is very reliable detecting the positive plants. The same thing happens with the other 2 primers, the B1 and the B8. As you can see, very reliable detection versus no false positive. That is essential. All 3 of them works very well. So we had to take a choice. So we chose to start with the A5. That is the one we have used for everything else. But by the way, we got 2 more sets in case they're needed, and they're again very, very reliable. So the first thing is to get the DNA extraction method that can be used in the field, and I think that everybody who is involved in this project will be aware of our developments. We have this scene, what we call the deep stick. There's a little bit of a handle with a little bit of a nucleic acid binding zone. It's pretty small. It's around 4 to 5 centimeters. By the way, it's all published. Those of you who are not familiar with it, you can see our paper here. And by the way, if you look at the numbers, as for today, it had 137,000 views. I didn't know there were so many scientists in the world, but obviously there are so many scientists. The basic principle that is in this paper is, you got your samples, you disrupt them, by checking them with a couple of ball bearings. Then you just dip the dipstick into the solution, you wash it, and then you just put it into your amplification tube. That essentially takes around 30 seconds, a bit more sometimes, a bit less sometimes, all right? That's okay. But for the development of the DNA extraction methods, that we had a number of goals. Obviously, it needs to be simple. It needs to be fast. We need to minimize the number of steps. That is essential. You don't want to have to do a lot of things when you are in the field, all right? It needs to be cheap. And another very important bit, you need to minimize the risk of cross-contamination. And that goes a lot farther than just what I show you or what is in the paper. Because, we need them to start optimizing everything, the methods that we're going to use. To start with, what kind of buffers are we going to use? What kind of buffer we're going to use for the extraction and for the wash, all right? We need to optimize the tissue amounts. For some diseases, some plants, you need quite a lot of tissue. For some others, you need very little tissue. And we need to optimize the maceration techniques. And you need to think of everything, how are you going to disrupt the tissue? Can we use ball bearings? Perhaps, if we put sand, is that enough? What kind of tubes are we going to use? We need, 50 middle tubes or 50 mil tubes, or we need only 15, or we need 1 mil tubes. You need to optimize all that. You even need to think, how long do you need to shake? If you're shaking, tissue with a ball bearings, is that 10 seconds? Is that 30 seconds? How much do you need? Because every plant, every tissue is different, and you need a different amount of time shaking them, to disrupt the tissue. Okay, well, we started with the tubes. That is what we knew, and we had at the beginning. And you see the ball bearings here, a couple of ball bearings in here. This is a 2 mil tubes. These are 2 mil tubes, and you can see that the different amounts of time shaking will give you different colors. That means that you are breaking the leaf in more pieces, and you're extracting a lot more juice and DNA into solution out of the leaves. But when you think about it, these tubes are, well, we have it by the hundreds in my lab, but they're not the best ones. They are a bit expensive. Also, putting things into the tube, if you need a lot of tissue, putting into the tube and breaking them into pieces, you need to do it with your hands. It takes time. The ball bearings are expensive. Of course, you can wash them. By the way, all our international partners with other projects, they wash the ball bearings. But we came up with an idea. Why do you need tubes? What about disposable bags? Can we just put tissue and the extraction buffer in a disposable bag and just use our fingers, to get something out of it? There are two big differences. First of all, bags are disposable. Tubes, you can wash them, all right? But, every time you wash anything when you're using amplification, you run the risk of contamination, all right? Bags, you can get rid of them, all right? And the other thing is the cost. These tubes are, 30 cents per tube or something like that. Ziploc bags, I just go into eBay. You can buy 1,000 of them for $28. That means that it's 3 cents each bag, less than 3 cents per bag. And that is in eBay and Ziploc bags. The truth is that you don't need Ziploc bags. You can use much simpler bags. You don't need to close them. So essentially, we've gone from a tube that you need to wash to something that is disposable, all right? And we tried it. So what did we try? When you start using the bags and you use your fingers to disrupt the leaves, and these are just pictures, for us, after trying to macerate the leaves in it. This is what you get, and you don't see much color here. Means it's not a lot of extraction that you get out there. But if you put a little bit of sand, you can see the sand here at the corner. You put a little bit of sand, that helps you a lot. And you can see by the color. I can magnify, the liquid of each of these two methods. This is without sand. This is quite transparent, and this is with sand that you can see that it got a yellowish, greenish kind of color. Means that it's working better. You are macerating your sample better. So we started working with sand, and we optimized a number of things for sand. One of them is, for example, how long do you need to, use your fingers, to break the tissue in the bags. This is just, relative amounts of extracted DNA. So we went from nothing. Obviously, you don't extract anything when you don't break the tissue. 5 seconds, 10 seconds, all the way to 25. And you can see that obviously the longer you macerate the tissue, the more DNA you extract from there. All right, to a point where you just don't really start getting any more. Around 20 seconds, that's it. Now, what is important, and we noticed that it's not an obvious thing. So, oh well, then 20 seconds it is. It's not that easy, because the more you macerate, the more DNA you get, but also you get more contaminants into solution. And then you're going to need to remove those contaminants in order to amplify, all right? Then we thought about something that we've used for other things, and that is diatomaceous earth. That, again, is super cheap. I mean, we have a container, I don't know, we had it for 20 years. It's like 5 kilos. I'm still, almost full. And we've been using it for 20 years, all right? Now, when we compared it, all right, this is the DNA concentration, all right? We found out that 50 milligrams, 100 and 200 of earth versus sand, 200, 300, and 400. And you can see that 50 milligrams is it gives us quite an improvement over nothing, all right? Something that is comparable to some of the somethings. And the problem with sand is that sand is very heavy, so it falls to the bottom of the tube. Means that you need to keep going to the bottom, getting the sand up, to wherever the leaf is, and rubbing the leaf. And when you stop the sand, little bit, every time you do it, the sand all goes to the bottom end. While DE, diatomaceous earth, stays in solution, so you don't need to worry about that. That saves quite a lot of time. So we found out that DE was the best way to go. And when you look at the DE versus nothing, and this is in blue, you can see nothing, and in orange, you can see 50 milligrams per mil of DE into the extraction buffer. And these are the percentage. We did dilution series, all right? So what you have here is dilution series 1.1, 0.01, 0.001 is a 10 times dilution series still, and the non-template control means there is nothing there. And what happens is you have very good reproducibility with both of them, but after 0.001 dilutions on all the other ones, when you use DE, you get around 66% of your reactions they're still able to find it. You don't get 100% like with the other ones, but that means that with DE, you're getting a much better reproducibility when you have very small amounts of material in there. So the entire process now is you macerate the tissue in a plastic bag. This is just a normal Coles plastic bag, all right? And then you use your dipstick. You introduce it into your extraction buffer that contains everything. Then you wash it, and then you put it into your amplification reaction. That is the entire process. Total, I would say around a minute, because the bit that you need to do with your fingers, on the bag, takes a bit longer than doing any other way, but it's not too hard, and you don't really need to push really too hard. By the way, you don't need to have a solution that looks green, all right? Even when they are almost transparent, that is good enough to detect things. So you don't need to kill your fingers, trying to disrupt the tissue. And then we go to the diagnostic droid. Again, you are aware of it. It's this one is the one that many of you know, the first version that we have of it. It's a portable amplification device. It's made into a coffee cup. This particular one would connect to your phone, all right? And you connect it with the phone. It's got a block inside to put 8 samples. It records interprets the data from the reaction. you can write it from a car battery or from a mains power, all right? And it can be used either in the lab or you can use it in the field. We use it in both places. That was the first version. The one that, you know, people involved in the project have seen now is this one. This got a number of improvements. The first one is that we have integrated in the circuitry some artificial intelligence, meaning that the machine actually learn how to interpret the data. The more, you know, we've done hundreds of reactions on it in a way that the machine knows what to expect. That is a lot better now. And it got 3 difference versus the other one, 3 improvements I think are really important. First one, it's got a heated lid. Heated lid is important because that way your reagents don't evaporate. It stays in the tube and it stays at the bottom of the tube. The second one you can see here is now it's got an LED screen. So now you can see all the results and all the communication. You can see it in the machine itself. You don't need your phone. This machine doesn't talk to your phone anymore. This machine doesn't need to talk to your phone anymore. So the app that we produce, now we don't use it because it's all here. And finally, the third improvement is this one. This is essential. This is, it was complicated to introduce this, but it's worth it. This is an SD card, meaning that all the data from every tube in every reaction is recorded here. And then you can take this, put it on your computer, and you can, of course, the machine will tell you the results, positive, negative, or inconclusive. But then you can look it up in your computer. If you have any questions, the previous one, you couldn't see anything. You have to trust what the machine was telling you. This one, you don't need to trust the machine. You can put it in your computer and you can check everything. And sometimes we've noticed that that is important. So the optimization has not only been on the outside, the optimization has also been in the inside. This is the first version. As you can see, there are a bunch of different circuit boards, one on top of the other. It was quite complicated, you know. Our version that now our partners are using, the one that we have sent to our partners, as you can see, it's a lot simpler. When you look at this and this, it's a lot simpler. It's got one, this circuit board has been designed, and we actually have someone helping us produce a design that we can now manufacture. So this whole thing is now, we can ask somebody to manufacture it for us. Instead of soldering every single component, we can order it now. Having said that, this is still quite time consuming to put together, all right? So we are now working in version 3.0. And what we've done is we have engaged a design and development company, a bunch of professionals. We're doing our best, but I would like to have the opinion of a professional. As we speak now, there is an electronic engineer having a look at our design. So what I'm asking of the engineer is like, can you please come back to me and say, Jimmy, that's the best you can do. Or Jimmy, you can do even better than that. All right. And finally, we will ask these people to redesign the whole thing to make it a lot easier to assemble. Now it's still complicated. I mean, it's not complicated. It's, well, look at this and this. It's a lot easier to assemble, but it takes time, and time is money. All right. So they have told me that they can end up giving us a design that you just need to click, click, and click, and that's it. You have that set up. So that's what we're working now. The next thing that we needed to look for is we need to send things overseas. Before COVID, they could go pretty fast. With COVID, they didn't go that fast. All right. Plus, even, you know, if it go pretty fast, you know, it would be fantastic to eliminate cold storage, you know, for reagents. If we ever want to produce kits that we send to very specific location in Southeast Asia that might not have a cold storage chain, or it could be complicated to keep things cold. So the first thing is we optimize the freeze drive for the LAMP reagents. We look everywhere. We couldn't find any available, reliable method to do that for LAMP enzymes. We've found it for PCR enzymes and other things, but not for LAMP. So we need to work on it, but it's okay. We did quite a lot of work on it. Now we are able to freeze dry the reagents. And of course, what we did afterwards is, keeping at room temperature for a while and then rehydrate the reagents and check the reactions, and they work okay. The activity is good. Activity is not 100%, but the activity is good enough. It's more than 90% what it used to be on fresh reagents. And now we have produced what we think is the optimal configuration. The diagnostic Droid have a capacity for 8 tubes. So what we do is we freeze dry 8.5 reactions to give a bit of extra volume. So every tube that we send in our kit has enough for 8.5 reactions. Now, those can be sent at room temperature. We already done it, and we've seen that it works because our partners have tried them. And then what our partners can do is rehydrate them whenever they need it, at the day that they can use it. Now, I think that I'm going to need 8 samples or 16 samples. Well, if I need 16 samples, I get 2 of those tubes and add the reagents. I mean, the rehydration solution that we also send to our partners. I mean, there is something that's left over. If you put it in the fridge, you can use it for a couple of days. So let's go to the technology transfer. The technology transfer, we sent the droids, by the way, the droids took forever. We were in contact with Jono, and it looks that they flew from here to Singapore pretty fast. And then in Singapore, they sat there for almost a month or 2 months. COVID is a humongous disruption in this kind of projects, all right? So, but the droids got there, and also sent in our reagent kits to do quite a lot of reactions, by our partners, all right? And we produce a set of very detailed instructions, protocols, all right? How to prepare the reactions, how to prepare and the sample, how to extract the DNA. And you can see here, there are documents in which the method is very detailed, all right? And we also produce documents, instructions on how to use and how to interpret the data on the diagnostic Droid. Everything that goes into that SD card has a meaning. We are produce this. So our partners, if they want to look at the data in their laptop, in their computers, they can do it. All those documents have been produced and have been sent. And then we also produce video material for training of the partners. We can have a look at the video that is here. See, after the sample is macerated, you can see that we just touch the dip stick into it, then we wash it. And then we elute the DNA into the tube. And that is the tube that we actually position in our diagnostic Droid. You have to excuse me, the video actually works, but all right, so there it is. So you put it into the Droid. and you get the results. Okay, and of course, you can always double check the results. You can always double check the results in your laptop and look at the different, things that are the different data that's been recorded. Okay. All right. So the 3D transfer, we did quite a lot of talk with our partners. All right. We were supposed to go and train the collaborators in person, but then COVID happened, so we had to do with the training video and a number of, let's call it, creative solutions. All right. We did review the initial results. All right. At the beginning, this was actually the first curves that the first results that we got from our partners, and it was perfect. It was just beyond perfect. It was perfect. Now, life is not that perfect, because after that, things stop being perfect for whatever reasons. All of a sudden, some of the samples were amplifying, but not all of them. And the thing is, these 5 samples were symptomatic samples, but they came flat. Only one of them came, and then they repeated it, and now nothing came. So then started doing weird things. It's really difficult to work when you can't really go there. Then Jono came to our rescue. He came out with the idea, said, look, I'm going to check, how things are doing. I'm going to record it and come to you. So thank you very much to Director Jono Spielberg. We can go to what he prepared, and it's all in YouTube, as you can see here. He did a fantastic job.
Copycat diagnostic droid. Let's go. Silly question. If you put more than one sample in the bag, there's no contamination.
Okay, as you can see, he did his best. I don't think that he has a particular, bright career in Hollywood so far, but if he keeps trying, he might end up, in Hollywood with a big villa in Los Angeles. Anyhow, that's what he's saying. That was fantastic. Actually, we watched that video, Jess and myself. It was an eye-opener for us, because we could actually then find out a number of problems that we could see there. All right. So we had some meetings to troubleshoot the problems. One of the things that we saw is that the last tube never worked. When we look at the last tube have very little volume compared with the others. So we assume that the P200 is way out of calibration. So even though we put enough reactions for 8.5, the 8th tube was never full. So one of the things that we figured out. Then we actually, after that, we had a live session. So we got Jono and his phone to have a live Zoom session while they were doing all the things that were doing the diagnostics. Again, that was fantastic. We could actually see everything and then get back to them. But aside from that, we did produce an additional document. As you can see, after our video meetings, we made a number of extra suggestions on things that we thought they were not quite all right. And after that, results have been quite satisfactory. Actually, we're pretty happy the way things are working. I think our partners are right on top of this, and they're doing quite well with these assays. So let's move to the witch's broom. Witch's broom is a completely different story. Witch's broom is a phytoplasmas. Phytoplasmas are not easy to detect. All right. There are very few reports in the literature for the cassava witch's broom, and there is very limited sequence information. There's some phylogenetic analysis, people who have amplified phytoplasma from infected plants, and then they've done phylogenetic analysis. Now, the interesting thing, that is scientifically interesting, but it's also practically interesting is I found different papers and they put it in different subgroups. And the truth is that, in my opinion, what I think might be happening is not that there is a dedicated phytoplasma that causes witch's broom, is that it has a lot to do with the geographical location. For example, I found in Brazil a group that have, isolated it, they got their DNA, they sequenced the DNA, and then they came up and say, oh, this is actually in the same group that this other Brazilian phytoplasma. This is in one of the papers that I read. So it's a phytoplasma that is in other crops in Brazil, and also they found it in cassava. A different thing, a Japanese group have published another paper with this cassava, which is broom. And the funny thing is that they find that when they sequence it, they found that it was almost identical to another phytoplasma that they already have in Japan. I think it's in citrus. So in my view, it might not be that there is one single phytoplasma that actually produces a witch's broom. It's that if there are phytoplasmas in the area, they can actually then jump from one crop into cassava. And in Brazil was this other one that they had, and I forgot, and I apologize, but I forgot which crop do they have that phytoplasma that jump into cassava. And in Japan was one that I think it was in citrus and jump into cassava. And that is important because that actually means that in order to be able to develop something for the one that we have in Southeast Asia, we need to be very sure of what is it that they are getting infected with. So we tried the published LAMP and PCR methods. They were, by the way, there is one group that actually has to do 3 PCRs, nested PCRs in a row. Mike, I have never, ever heard of doing 3 nested PCRs in a row. So what did we get? We found inconsistent results. The LAMP primers that have been published, they don't work well for us, for a number of reasons. One could be that if those primers were developed for whichever cassava phytoplasma they have in Brazil, they might have been developed for the Brazilian phytoplasma. They might be a different one that we have in Southeast Asia. Again, I don't have proof of this, but I got a strong suspicion that could be happening. So yes, sometimes we were able to detect it, and then we would repeat the reaction, and then we were able to detect it. So we found problems, all right? One of the problems I can tell you, and that is even with the published protocols, is that they can amplify cassava DNA. And this is the proof of it. When we look at the genomic region in which we're able to amplify, we found that in cassava, this ribosomal interspaces region are very similar, all right? And we found that not only they're very similar, but there is one difference. In cassava, that particular amplified region have an MBO1 side, a restriction side, an MBO1 side. Then we did one very interesting experiment. We did a PCR from cassava plants infected, with witch's broom. Here it is, beautiful band, great. That's the PCR, all right? Now, if you digest this band with MBO1 that is not in the phytoplasma for cassava, but is in the cassava genome, you can see that the band gets digested. Equal one is not, so you get the same size of band, but this one gets digested. So what we are detecting here is cassava DNA. Then you can forget about these primers. These don't work at all. Now let's look at this. If you digest your genomic, your DNA before you do the PCR, you digest it with MBO1, the band is not there anymore. You can see a little bit, but it's almost not there. Means that you have destroyed the target. So what are we amplifying? We're amplifying cassava DNA. So where are we now? At this time, with the available sequence, we cannot, it's impossible to develop a reliable method. And let me explain this. We need sequence for the phytoplasma that we have in the region, not the sequences that have been published. And I'm saying that because remember that I told you that for whatever reason, people, are doing this genetic analysis of the witch's broom, and there is always suspicion very similar to other phytoplasmas that already exist in other crops in the region where they are working. So with this additional sequence information, Wilmer at CIAT is working on it. So we are on it. Sooner or later, we're going to get that information. It's difficult at this time because the quality of the DNA we have or Wilmer has obtained, it hasn't been really good. So, but we're working on it. As soon as we get more information, I'm pretty sure that we can come up with a solution. And now we got to the other thing that everybody's talking, cost of reagents. All right, so this is an analysis of cost of reagents. Jono asked us to do it, so we've done it. This is all the reagents that we're using. This is the manufacturer. This is the catalog number. This is the price for the amount that they deliver. And this is even the delivery they charge to UQ. So we put everything in there. When we put everything in there, there are two big numbers here. This is for 1,000 reactions. One reaction is too little. There are too many zeros for one reaction, all right? For 1,000 reactions, you look at it, there are two big items here, and that they are the enzyme, what is not surprising, and the DNTPs. That is actually surprising. I didn't know that was so expensive. So when we put it all together, it's $1,463 for 1,000 reactions. That comes to $1.46 per reaction. Now, $1.46 Australian dollars per reaction. I know that is expensive. Let me tell you one thing. Again, that is my own opinion. I know it's expensive, but it's relatively expensive. It's not that expensive when you think that when we have one of these projects and we send one of us, travel from Australia to Cambodia, you end up spending $4,000, trip, hotels, etc., etc. So those $4,000 pay for all of reactions. If we have to come from America, it's even more expensive. So I know it's expensive, but let's put things into focus. Again, that is my opinion. Now, let's analyze the cost. The enzyme is around 60% of it, per reaction 88 cents, and the DNTPs is 40%, per reaction 59 cents. Now, 60 and 40 is 100. Yes, the rest is almost nothing. The rest is just pretty much nothing. Okay. So let's go for the DNTPs. As I told you, we buy it from NEB. We buy it in a big tube that is cheaper than the small tubes. 40 micromoles of DNTPs cost $390. Now, what I've done is I spent a little bit of time finding these things in China. China is the new king of manufacturing for everything, for cars, for phones, but they've gotten into science, into reagents, into molecular biology, and they have a lot of manufacturers of molecular biology reagents. So I spent a little bit of time looking for it, and I found this company. It's called Shine Gene, in which you can buy the same amount that you buy in NEB. It costs $81, not $400, $81. And here it is. This is a tube, the NTP is €8. All right. I'm assuming that they're putting in euro because they are selling it to Europe. If they're selling it to Europe, it's because it works. All right. Otherwise, you wouldn't say it to Europe. Let me explain this. Okay. Now, one reaction NEB is 58.5 cents for DNTPs. One reaction with Shine Gene reagent is 12 cents. That actually comes up to 40% savings from getting it from an Australian company or an American company in Australia, or getting it from a Chinese company. Same thing. Now, if we extrapolate and we think that we can get the same 80% savings on the enzyme, and I have not been, I haven't had the time to look for it, but we can make that one of the things that we want to do, find Chinese manufacturers for all this. If we are able to get 80% savings on the enzyme, and I know that enzymes in China, they're selling them that there is no tomorrow. Like so many manufacturers of enzymes, you wouldn't believe it, okay? If we can keep these savings, our reaction will go from $1.50 to 30 cents. So there you go. I think there is a way to go, and we can do that. Of course, you know, we need to find, you know, the manufacturers, you know, we need to test that they work, etc., but it's something that can be done. So what are our incoming priorities for you know, the foreseeable future in this project? You know, we really want to do personal training of the panelists. That makes the whole difference. That would make everybody comfortable. They can ask questions. We can look at them. The university, my news are that they might allow to travel from July. Now we're not allowed to go anywhere, but they might allow travel, overseas travel from July. Now we're only allowed to travel in Australia. So that if that is allowed, that would be our priority number one. We really want to go. We're going to spend time with all the panelists. If it's necessary, make a workshop, or vice versa. We just jump from place to place and we spend 3, 4, 5 days in each place, or a week in each place, you know, working with the panelists, doing reactions, using the droids, using the deep sticks, extracting DNA and analyzing samples. We need to develop the which broom test that, you know, we need to do. We need more sequence info because what we have, I'm not sure that nobody can actually do anything reliable. We need to know what is the sequence of the specific phytoplasma that is associated with the Southeast Asian cassava population. And then different applications, then the needs development, you know, can we do pooling of samples to save even more money? If we want to do that, can we test stems? We can test roots. Yes, I can tell you now. Yes, we can test stems and we can test roots. We need to work on it. It's not optimized yet, but I can assure you we can do that. Reduce assay costs. As I say, you know, we can look at Chinese manufacturers. And by the way, those remember this. I spend the best part of 20 minutes looking on the internet. By the way, if you go into Alibaba now, you can actually buy reagents in Alibaba. And they are even cheaper than the ones you can buy in eBay. So we need to find the proper manufacturers. Again, I have excellent relationships with China and an excellent relationship with CADAS, the Chinese Academy of Tropical Agriculture. And we can actually, through them, we can source things. And then the final thing is we need to make these things last. So one possibility is commercialization. You know, that's one possibility. You know, what about, you know, deep sticks and like that? And by the way, I didn't tell you, but deep sticks now we have come to a way that we can produce thousands of them. So it's a lot easier than it's even published in the Nature Protocols papers. We can do, it's not automatic, but we can produce quite a lot. The other stuff, well, I mean, I'm open. You know, I actually had some discussions with CADAS before COVID happened on how could they actually manufacture both things, the Droid and the deep sticks. You know, that could be, you know, one of the solutions. You know, I don't know any other companies. Finally, and I finish my presentation here, I want to thank everybody, all international partners, from the people in Colombia, who's been fantastic, to obviously the people who are working every day in Laos, in Cambodia, in Vietnam, in Thailand. You know, they've been really, really good. And whenever we needed anything, they have prepared and sent it to us. So thank you very much, guys, for that. And thank you to Jono, because he's been extremely efficient. Every time we needed something, Jono would have everything ready almost in no time at all. And he's been very patient with us and very understanding that COVID is making things difficult. All right. So that's where we are. That's our presentation. I thank you very much for your attention, and I shall stop the recording now.